Okay. For those of you who are here today, nice job. Thank you. We just have about 50% attendance today, which is not great because I have a super important announcement about Wednesday's class. So I already talked to a couple of you about this, but if you have not taken your exam yet, please do so today. I'm in my office all afternoon. If that's super impossible for you, I can have you come to my lab and you can take it in the back of lab tomorrow, but I don't have any office time tomorrow. So talk to me after class if you need to take your exam. The reason it's important that you do so today or at the latest tomorrow is because Wednesday we're going to be redoing it as a small group. Okay, so you might not have done that before. Some of you did really well on it. I have graded a little bit over 50% of your exam, so I was working through those on Friday. Some of you did not do awesome. So there is some variance. Some of you who did well the first time didn't do quite as well this time, and some of you have improved your scores. So kind of an interesting distribution so far. That said, the way we're going to do it on Wednesday is in small groups, and I haven't decided the exact number. 6 would be too big, probably 3-ish people would be good. Maybe 4, there can be some variance there. But basically, it's going to be the exact same exam, so you've already seen those questions, right? I'll give you back your note sheet, and what you're going to do is work on it as a group. If your score so keep in mind, this is going to be a separate category. It's not going to replace your original grade, but there's going to be 25 additional points available for you to earn back, which will help for those of you who performed really poorly on it. I will also still consider a curve once I finish all of the exam grading, so that would be a separate issue, but this is going to be a way for you all to kind of reflect on the important content. One thing to note, if your score on the group exam is lower than the score you got on your original, you'll just get the score you got on your original exam for that second assignment, up to 100%. If you got over 100%, I'm not going to give you extra credit, but up to 100%. Questions on what that will look like? It's going to be 25 points. It'll be the exact same questions. Like I said, you can work on it in small groups, so you should be able to chat with each other. And the vision is that multiple brains are better than just one, so hopefully you can get an even higher score on that second portion. Like I said, I'll bring your note sheets so you can have those back. And that'll be on Wednesday. So think about who you want to work with. If you feel like you didn't study appropriately or some of the right content for the exam on Friday, think about what you might want to look at before you come on Wednesday. There's no class on Friday. I'm not going to be here. You guys don't have to be here. Your infographic is due at 11:59 p.m. that day. So there is some time, maybe in that morning, that you could work on it a little bit if you had not. So these are some important reminders. I am going to send an announcement on Blackboard about the activity on Wednesday. So just in case people aren't here. Yeah. No, so in Blackboard, you have 50 points for the test, and the score that you got is going to stay the way that it is, again, unless I haven't finished grading everybody's tests, but based on what the average class score is, and there's some people who haven't taken it quite yet either. So once I look at everybody's tests, if I add a couple of points to a curve, that would be separate. So you'll still have that column. What I'm going to do is create a new category that's going to be 25 points, and it's going to be this group exam. If you do the group exam and you get a higher score, it should do a good job to help boost your grade. If the group exam is lower than your original score, I will just replace it with the percentage you got in that first one. Does that make sense? Yeah, so it's not going to replace it, but it should help to kind of buffer your grade a little bit, provided that you can get a higher score on the group exam, which is typically what you see. Typically, we see this. Any other questions? And like I said, since you've already seen a lot of those questions, you might recognize okay, I didn't necessarily study osmosis, so I'm going to do that so the next time I can nail that question on where the water is going to go in or out of the fish, right? Because some of you crushed that and some of you did not crush that. So maybe relook at that. I had the exact example I put on the test in one of my PowerPoints, so you can go look at it before Wednesday. Check it out. I even used a parrotfish both times. We are going to get going into a new topic. Today is DNA structure and replication. We've talked a little bit about DNA in different capacities, but we're really going to be focusing on some of the important characteristics that the DNA molecule has that especially make it kind of unique and very interesting. And then we'll be talking about the replication process. I do have a short video embedded in today's lecture because it's honestly really helpful in visualizing some of these processes. A fair number of learning objectives. I'm going to briefly mention the history of DNA. We're going to talk about the structure and function. As always, there's some important enzymes involved with the DNA replication process. We're going to talk about those, and I want you to understand how the structure of DNA influences the replication. So, for example, there are anti-parallel strands, which is important to how that replication process occurs. So getting into the history of DNA, there's actually lots of different novels and books and nonfiction things that have been published about this. So there's a lot more you could get into if this is a topic that interests you. Back in 1860, there's a scientist, this man named Frederick Miescher, I think is how you say his name. He was working on something kind of gross. He was actually looking at pus in Band-Aids, which is kind of nasty. But one of the reasons why this was advantageous is because pus is essentially an immune response. And we know that immune responses are mediated by white blood cells, which do have nuclei. And this is how originally excuse me, originally we determined that nucleic acids existed, and the way that he did this was by looking at these white blood cells, looking at this pus, and then found that they tend to be an acidic substance. So this was quite a long time ago. This was a foundational discovery in terms of figuring out what DNA is. In 1900 and 1950, scientists didn't really know how heredity worked, and they didn't know what the heredity molecules were. Originally, they thought it might be proteins because there's about 20 amino acids, and they thought there seemed to be more possible combinations there than with just 4 bases, which is a good hypothesis. But as we know now, of course, proteins are not hereditary molecules. DNA is. In 1952, they did some interesting experiments with something called bacteriophages. Bacteriophages are really kind of crazy. They're very interesting. They're bacteria that infect viruses. Sorry, flip that. They're viruses that infect bacteria, excuse me. And basically, the way they use these bacteriophages is by tagging both proteins and DNA and then tracking how heredity worked. They were able to determine that it was DNA rather than proteins. This was done by some scientists named Hershey and Chase. And then just a couple years later, 1 or 2 years after that, the structure of the DNA molecule was published, and it was that double helix X structure. The folks who were working on this were Watson and Crick. You might have heard of these two. There is some contention here. There was a scientist, Rosalind Franklin, who made really important contributions with some of her photography. She was looking at DNA as well. They actually used some of her data without her permission and then published on it and got a lot of the credit. So this is, again, something you can read about more, but very brief history on DNA. Now, this should be a review. What is DNA? It's the heritable material. Remember, it's like the instructions for all living organisms. It's a nucleic acid. It's a polymer. These are all words we've talked about before. Okay, here's your quiz. What is DNA made up of? Sure, say it. Yeah. Deoxyribonucleic acid is what DNA stands for, but we do have nucleotides, which are close. And we know that nucleotides are made up of those deoxyribose sugars, which is why it has the name deoxyribonucleic acid. We also have these important other components, the nitrogenous bases and the phosphate groups. So I've taken this figure here and I've included some important annotations with the labels and the pink arrows. The circle is showing what a nucleotide is comprised of. So remember, nucleotides have all 3 of these components, a specific nitrogenous base, a phosphate group, and a deoxyribose sugar. And for DNA, we have 4 different possible nitrogenous bases: adenine, thymine, guanine, and cytosine. So when you look at this figure, we have pink and blue, and it looks like purple is the last color on here. So these are all representing our different nitrogenous bases based on color. Now, the different groups combine together to form what we call polynucleotide chains. So these would be like the polymers, and then the monomers are these nucleotides. So the nucleotides are the monomers, and they combine together to form these polynucleotide chains. Now, something that's kind of cool is there's a special type of bond called a phosphodiester bond, and this is basically just a covalent bond, so it's strong. So we have a backbone of DNA. It's what we refer to as these links. These phosphodiester bonds form the backbone. And then in between these two backbones, which are shown here in light blue, we have our bases that are linked together by hydrogen bonds. So we have lots of different important bond structures involved in the structure of DNA. Now, the chain has polarity. Will you all remind me, what does it mean to have polarity? Sure. Yeah, absolutely. So we have like slight differences in the charge. We have positive and negative ends. And the way DNA is labeled is according to this polarity. So we have 5' and 3' ends. And the little dash, you might come across this in your travels. You'll see them up here, they're abbreviated. We have the 3 and then the little dash mark. That's pronounced 3'. So this is the 3' end, and then this is the 5' end. And I have another slide that looks a little bit closer at that here in a sec. But basically, the polarity of the DNA molecule allows for it to be both stable and functional. So when I say functional, it helps transmit the important genetic material and the important information to be able to make proteins, etc. So we have these deoxyribose sugars and these phosphates linking up, forming these phosphodiester bonds, making a really strong backbone. And then in between those backbones, like the rungs on a ladder, we can think of DNA a little bit like a ladder. These are the outside, and then the rungs are the bases being linked together by hydrogen bonds. Does anyone have any questions so far? And just to be clear, in terms of like what parts of this molecule might be more capable of interacting with water or in aqueous environments, generally we would expect the backbone or these deoxyribose sugars and phosphate groups to be able to interact with water, and then this internal would be a little bit more hydrophobic. As promised, just a bit more information here. You don't have to, this looks kind of scary, but there's nothing super crazy going on here. This is showing the way that these bonds or these important covalent bonds are formed, and the different nucleotides are basically just color coded. So you can see here we have 3 different nucleotides, and the different 5' and 3' are associated with carbons within these molecules. So a couple more components about DNA. It's a double helix. You might have heard this before. And that just means rather than having one polynucleotide chain, so here we are looking at 2 of those polynucleotide chains. So when we look at this would be one, and then this would be another one. So DNA is comprised of 2, those polynucleotide chains, which gives it this double helix structure. And they actually wind up so that they're kind of in this spiral staircase looking structure. As I was saying, that sugar phosphate backbone is on the outside, and then those bases are on the inside. And this isn't important, you don't have to really know this, but I think it's kind of interesting. DNA usually coils into a right-handed helix, so I found this figure to kind of represent that. A right-handed helix curves upward in the direction of the fingers of the right hand when your thumb points upward and your fingers point inward. So you could do that, and you could see the direction that DNA is usually going to spiral. Something else to note about DNA, there's certain areas where more of those internal bases are shown, and there's areas where fewer are shown, just based on the way that it essentially tends to associate. We have major and minor grooves. Major grooves are bigger, they have more nucleotides showing, and minor grooves are smaller. That's why it's called minor. They have fewer base pairs showing. And basically all grooves expose different base pairs. Now, the reason this can be important is so other molecules that might need to interact with DNA can identify specific locations in the genome by binding to certain nucleotides. So these groups facilitate those interactions and they allow the specific sequences to be recognized. Hey, as I was mentioning at the beginning for one of our learning objectives, DNA is anti-parallel, which means our two polynucleotide chains are going to be opposite directions. That's all the anti-parallel means. Now, the reason we see this anti-parallel arrangement is because of complementary base pairing rules, which essentially means there's limited amounts of space between those sugar phosphate backbones. There's not an unlimited number of spaces or amount of space. And within the available bases, there's some that are slightly larger and some that are slightly smaller, and we can only have one big and one small matched up together. Same deal here. This anti-parallel structure is emphasized when we look at the 5' and the 3' opposite ends. Now, in thinking about like what this actually looks like, the 5' end has a free phosphate group, which you can see when you look up here on this relatively blurry picture. We have this P representing a phosphate group, and then we can see down here, the 3' end has this hydroxyl group or this OH group. Now, it's not super critical that you know that, but it does matter in terms of DNA replication. The different 5' and 3' ends have different like molecules attaching to them, and certain operations can only be facilitated on either the 5' or the 3' end. So it does matter in terms of the function of DNA, but I'm not going to be asking you like which of the important chemical groups is on the 5' end. It's not super critical in that sense. Questions here? Okay. Again, this figure looks kind of scary, but I just wanted to talk a little bit more about some of the naming schema here. Same deal, I've circled the molecules that they're looking at. So the 5' and the 3' labels refer to the numbers assigned to the carbons in the ring. So that's the magenta number. So you can see that it's talking about which molecule is bonded where. And we call it the 5' because we're talking about the phosphate group being bonded to this 5 sugar or the 5 carbon in this sugar. And then same deal over here, the 3' is being bonded to that hydroxyl group. So that's where the naming comes from. As I was mentioning, there's a couple of different types of nitrogenous bases. We have the smaller ones, which are our pyrimidines, and then the larger ones are purines. So a main difference between these groups of nitrogenous bases is whether there's one carbon-nitrogen ring or two. Two rings makes them bigger, one ring makes them smaller, of course. Now, this figure, and this is pulled from your textbook, is where I got this. We do also see uracil. It's important to note uracil is not present in DNA. Uracil is an important nitrogenous base, but it's only present in RNA. So in terms of nitrogenous bases, we're going to see in DNA, we're only going to see cytosine, thymine, adenine, and guanine. Does that make sense? Sound good? RNA is a separate type of molecule. It is comprised of nitrogenous bases, but RNA generally only has one strand and it has a wide variety of different functions, whereas DNA tends to pretty much function in the exact same way. It's important for storing the genetic material, storing the hereditary information, and then helping to make proteins, whereas RNA is widely involved in the process, which we'll talk about more later. Now, the way that these different molecules or these different nitrogenous bases differ is mostly by the chemical groups attached to the rings. So if you note, you can see like an NH3 or an NH2, there might be an ammonia group or an amino group attached, and that creates their differences essentially. So again, just to reiterate here, between the sugar phosphate backbones, there's limited amounts of space. So there's always going to be one pyrimidine and one purine. One big, one small, linked up with hydrogen bonds. And you can see those hydrogen bonds here, which I'm showing again with these pink arrows. Interestingly, between certain bases, so between guanine and cytosine, there's actually going to be 3 hydrogen bonds, whereas between adenine and thymine, we actually only see 2. Now, the reason this happens is largely because of the shapes of these molecules. And as I was mentioning previously, this is what we refer to as complementary base pair matching. The really key term here is complementary. If we know one strand of DNA, based on complementary base pair rules, we know the other. This is because cytosine and guanine are always going to match together, and thymine and adenine are always going to match together. So if we know one strand, we can say the other. Questions here? This figure is just to kind of reemphasize that point. You can see at the bottom, I have like a couple of strands. This figure is showing strands of DNA. And if I covered one of these up, you could tell me the other in theory. And if I covered this one up, you could tell me the opposite, again, based on our complementary base pairing. So we'll practice this next class. I'll probably give you a little example of trying to come up with the complementary strand of DNA if I give you 1/2. This is like a really classic test question you come across in like a lot of general biology classes, like tell me the complementary base or tell me the complementary strand of DNA given this template strand. So do you feel like you could do that based on these base pairing rules? A with T, C with G? Okay, I should have given you a practice here, but instead I gave you Sudoku for your break. So work on this, solve our Sudoku puzzle. If you need a review on the rules, let me know. Got it done, Celine? Are you ready for the answer? Do you need more time? Ready? Is everyone else ready? You can check your work. Is this what you got? Check out your answers. You can see. Okay, we're going to move into our next set of learning objectives here now. So focusing in on the DNA replication. As promised, I do have a video for you, so hopefully it will play here. This video is great. It's only like a couple of minutes long. I do ask that you take notes on what you're seeing, and then at the end, I'm going to have you discuss with someone near you what some of the key ideas were. So keep in mind, it's not nap time. This is focus time. And I might have to adjust the volume, so.
DNA is a molecule made up.
Is this big enough, or do you need me to make it bigger? Let me make it bigger. Or do you think it's okay?
Two strands.
Okay.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay. So, like I said, discuss with someone near you what the main or the most important parts that you thought were. Maybe you would focus on some of the key enzymes. There was lots.
Probably forgetting some things and you can add.
It goes from, top one, I guess, it goes from 500 to 500.
The old like so like when it's broken apart, you mean? They're both used as template strands for the new ones. So according to the complementary base pair matching, that's how the new strand is built. So as the DNA polymerase, that molecule that adds the nucleotides, sees like a T, for example, it knows, okay, I'm going to add an A, and then it just keeps moving down the line and adding the complementary base. So they stay there. So one piece of DNA is going to serve as a template for two new ones, and then you're going to get like two pieces, which would have 4 strands that are combined into 2 helices, and then same deal. Those could open up, and then you could have more. Does that make sense?
Primates.
Hey, I think I heard lots of great things here, which is excellent. Let me find my mouse. We are going to kind of go over, hopefully, come on, some of the important components here and add a little bit of context as well. So remember, DNA replication happens at a certain time in the cell cycle. Can anyone remind me where it happens in the cell cycle? The S phase, yep. So during the DNA synthesis phase, of course, and this is going to be before the cell goes into any sort of nuclear division. So it's going to double the amount of DNA. And this is basically the process of how that DNA is copied in the first place. So that's kind of what we're talking about here. Now, as that video was mentioning, the process of DNA replication is semi-conservative. Now, the reason that we specify this is because for a while, and if you wanted to, I believe in your textbook, if not in many other textbooks, there were other kind of hypotheses for how DNA replication might be occurring. And there were some that thought it was a conservative model or like an entirely new model. But basically, we call it semi-conservative because each new piece of DNA is made up of one old and one new, which is why we basically emphasize this. The process overall, of course, requires many different enzymes. So all of those molecules that were shown with like big brown blobs, oh my gosh, ending in ACE, those were enzymes. So very important to this process. Now, DNA replication is fairly quick. The entire human genome can actually be replicated in just a few hours, which is kind of crazy because that's a lot of different nucleotides. It's also very, very accurate. We say there's only like 1 in 10 billion errors. So let me make that a little bit more clear. Like if there's going to be 10 billion nucleotides replicated, we would expect only there to be an error with one of those nucleotides. So it's super duper accurate and very quick, which is why this is a cool process. I don't think that today we'll have time to talk about how it fixes errors, but in the PowerPoint I have on Blackboard, the last 2 slides talk about basically fixing issues when this process doesn't go exactly to plan. Now, of course, it's very important that this process is accurate because if it's not, we might not get the proper amino acid combination, and the proteins that are formed later from this genetic material or from this DNA, they might not function properly. So we're very grateful. Thank you, DNA replication process, that this works the way that it does. Did you have a question?
Cancers get formed, it's when DNA replication doesn't like go well and then the cells are all messed up.
Yeah, so I mean, a source of mutation, which is what we would generally consider that to be, could absolutely be from this. Although in general, the DNA replication process is so accurate, it's not like a meaningful source of error. But yeah, it could be a contributing factor. The big issue would be if that happened one time and then that piece of DNA had an error and that piece of DNA continued to get copied, we could see more over time. So yeah, that's a great thought. Any other questions? Cool. So as we were kind of just reviewing, the DNA replication process starts first when DNA is unwound, and it's also opened at a specific site called the origin of replication. So the video doesn't do a great job of talking about this, but essentially there's a specific sequence of DNA that signals the other important proteins. So before any of the replication can even occur, we have to open up the strand of DNA. And if you remember, the DNA is bound together or held together basically by hydrogen bonds between those different bases. So the hydrogen bonds have to be broken essentially. DNA helicase does important work of unwinding the DNA and forming something called a replication fork. And on the next slide, I have an interesting figure of that. Something that's also not mentioned in the video, these are important. They're called single-stranded binding proteins, and basically they coat the exposed piece of DNA and keep it from joining back to the other one. So the DNA gets opened up and then single-stranded binding proteins are applied so that it doesn't just reform that double helix. Now, interestingly, eukaryotic organisms have tons of different origins of replication, something like 30 to 50,000 different origins of replication in the genome. So the reason that this is able to happen so quickly is because it's not just happening in one place and then kind of moving slowly, it's happening all over at one time. Related to that idea, DNA replication is occurring in both directions, so we get these replication bubbles. How do the origins of replication stick back together? So the origins are basically just a specific sequence of DNA. So that is essentially why it's not like a separate molecule. It's just the location where other proteins identify they need to attach. So when I was talking about like major and minor grooves, for example, there's certain pieces of DNA that allow other molecules to recognize where they need to attach. And so it's related to that idea. Mm-hmm. Yes, so the breaking is a separate process, and the breaking is done by a different enzyme called topoisomerase. So the opening is a separate process from the breaking. And what's kind of crazy is the molecule, and I'll talk about this in 2 slides from now, the molecule that breaks the DNA actually kind of helps manage the force of the twisting strand. But does that answer your question? So like if your piece of DNA is like this and a chunk gets taken off, it's basically like flipped around and retwisted. And then how exactly does it get back attached? It just flips it in motion in time. And then ligase seals up the gaps later. Sure.
How is that point of or like origin of replication chosen?
How is it chosen? Well, I'm not sure if I understand fully the question. Like over evolutionary history, we would expect there to be like specific locations that were selected, but the origin of replication is specific. So it's going to be the same every time, and it's a specific sequence of DNA. So like CCGT or whatever it would be.
So it just depends on the DNA.
Yes, it depends on the organism. So it might be different for other eukaryotic organisms. Yeah. Any other questions? Okay, so a little bit closer look here at some of these ideas. You can see the dots at the top of this figure are representing the origins of replication. Of course, when we look at this, we see DNA not replicated, represented in a lot of different ways in various figures. So sometimes it's as simple as just like a line, which is what we're seeing here. So each of these blue lines represents DNA. And so we have 2 strands, these origins of replications, and these are where we're getting our replication bubbles. And then the fork is basically this component where it's splitting apart. That's why it's called a fork. So those are going to proceed, and that is when this process is complete. The bubbles are going to get bigger. They're going to basically kind of stretch out. And we can see some of those same ideas here. The little gray ovals are our single-stranded binding proteins. So remember, those are the molecules that help keep the DNA from just like smacking back together, from hooking back together here. So we have those SSBs on both of these strands in order to keep the DNA separated. We also have our replication fork. Some other important molecules here that we're going to be talking about on the next slide. We have our primase, which is going to add primers. We have topoisomerase, which is one of the important molecules for this kind of breaking process. And then we also have helicase, which is physically breaking apart the strands of DNA. So you can see here, topoisomerase is going to break, swivel, and rejoin parental DNA ahead of the replication fork. This is crazy cool, relieving the strain caused by unwinding. So topoisomerase is actually able to kind of mitigate some of those pressures, which I think is kind of cool. Questions here? Okay. Part of the way this process proceeds, as I was saying, topoisomerase prevents the twisting. Primase makes primers. And as we talked about in lab, primers are little bits of RNA. They're important nucleotide segments that help polymerases form DNA. Now, polymerases are awesome molecules. They're very good at adding nucleotides in a specific order, but they actually have no idea where to attach without the primers. That's why we have to have primers, because otherwise the polymerase does not know where to make that DNA molecule. As the video mentioned, DNA is always going to be synthesized in the 5 to 3' end, so nucleotides are being added to that 3' end. Now, because DNA is anti-parallel, only one new strand is made continuously, and as a result, we get leading and lagging strands. Now, the lagging strand is the one that's made discontinuously or not continuously, and the leading strand is made continuously. And the video did mention this keyword, the Okazaki fragments. This is like, I don't know, Trivial Pursuit question. They're named after the scientist who discovered them. That's why they're called that. His name was Okazaki. So some of those key ideas are shown here. I added annotations again for you, lagging and leading, you can see. So our leading strand is complete and it doesn't have to be basically zipped up by ligase, but this discontinuous or lagging strand made of these different fragments does have to be basically patched up at the end. Now, the reason that we get this process is, again, because of the anti-parallel structure of DNA. Does that make sense? This is another figure. I like some of the key components here. This is also showing some of the free nucleotides, which are basically floating around in cellular space that are being added to the new DNA strand. And again, multiple DNA polymerases are going to be working at the same time. So in this figure, the polymerases are those big blue blobs. And this is kind of showing how this process is going to conclude. So the leading strand, as I was talking about, synthesized in the direction of unwinding. Lagging strand synthesized in the opposite direction. Now this important molecule called DNA ligase is going to seal the gaps. So this is kind of hard to tell, so I made arrows here as well. This figure is showing, okay, this is a gap and this is a gap, and it's saying ligase is basically going to seal those gaps up. That's what its job is. As I was mentioning earlier, those replication forks from different origins are going to move in opposite directions. The bubbles are going to get bigger and expand until eventually they meet and the DNA molecule will have been fully replicated, which you can see in this figure, that replication bubble expanding and stretching essentially. And this will keep going and going. So maybe I'm misunderstanding your question. The DNA is opened and then the molecules get inside there, but it does actually get broken by topoisomerase. It gets broken and partially handles the untwisting process. But it's not like chopped up into pieces. It's opened and then chopped a little bit inside once it's opened. I can talk to you more after class. Yeah.
Is there an origin where it starts? Is there a place where it stops?
It basically stops when it reaches the end of the strand of DNA.
That's trying to replicate?
Yeah, good question. Yeah, so it stops when they meet the other replication bubbles as well. Can be an alternative. Okay, I do have a little review activity here. This can be kind of fun. I want you to be creative here. So with a partner, I want you to come up with nicknames and short descriptions for each of the important enzymes. So the first step here is going to be defining the important enzymes. And I do want you to be creative with the names. There's a couple of key enzymes. Maybe write these down. This is your task for the next couple of minutes. This is good for helping you remember what these different enzymes are doing. Okay, let me try to understand your question better here, Celine.
But it's just chopped in between the strands to open.
Yes.
Just open.
Yes.
Not.
It is chopped a little bit. So that big, if you go back a couple of slides, keep going a little more. This molecule, topoisomerase, actually does cut out little chunks of DNA, but like I realize now where I was being confusing. So you're asking if it gets cut like this, and topoisomerase is the only molecule that does actually do some of that cutting. It basically cuts that piece, handles the pressure from that twisting, and then like re-sticks it back in there. So that's the only time it gets like chopped. But when it's open, it basically just gets like separated. The proteins coat it, and then the bubbles go. Does that make sense? Okay. I was like, I am not understanding. We must get to the solution here.
Primase. Synthesizer.
No, we need to know what it's called.
Which one?
The one that unzips.
Helicase unzips.
Helicase, he did.
They all what? Because they're so important. I don't know. I feel like the SSBs definitely are not as important as like polymerase. As LeBron, yeah. Also that. You could find like a less important basketball player for them. Yeah, go through, give them all names. Yeah, I mean, that's creative. You could give them names of players based on their roles. When you finish, hang out for a sec, and then I probably will have time to talk about DNA repair. Also, make sure you review the beginning of this lecture because we're going to do a group exam on Wednesday. Did you hear about that? Did Augustus tell you?
I looked at the slides.
Okay. It's not a huge, I guess it is matters. You want to be there, but it's not a huge deal.
Yeah.
Did you get some fun nicknames? Not very smart. Yeah, it is weird, right? It's like, it's so cool that it can do that, but yeah, it's like, I can't do anything else. And it also needs the primers or else it couldn't attach anywhere. Yeah. So remember when you were talking about PCR, there's Taq polymerase, which is just a different type of polymerase. It's doing the same thing.
With bacteria and like the hot springs.
Thermophilus aquaticus, I believe, is its scientific name, which is why it gets called Taq.
Remember that from last year?
Did you talk about this last year?
Yeah, we did a lot of like PCR.
Cool. Well, we do have a DNA technologies lecture, not next time, but I think the time after. So we'll talk more about some DNA technologies. They are cool. There's lots of methods. PCR, CRISPR. I'm going to make you debate CRISPR.
Our syllabus put back another one because of the testing on Wednesday.
Because of me having to let everybody redo the test. Is it theoretically sort of out of sync now? Yes. But I do just have to adapt to like what the student need is. So if everybody had passed the test and done a great job, we could have been like, great, we'll keep going forward. Unfortunately, that was not what I witnessed.
Do we have to test again?
Do you have to?
Like, I'm pretty sure I did. I don't like, I'm pretty good.
That's a good question. Do you have, if you feel really confident, do you have to?
I don't want to be cocky here.
Yeah, you don't even know your grade yet. That's a good question. Let me think about it for a second and I'll tell you at the end of class. Or maybe I'll post it in the announcement. Let me think. What are you saying? Funny names? 3. There's definitely more than 3 important enzymes, but okay.
I thought there was 3 in that video.
Well, what about the lecture? I gave you lots. Okay, what 3 did you talk about? You can tell me those.
DNA polymers.
Okay.
Primate.
The DNA polymerase,
Yeah.
primase, and helicase. Okay, what names did you give them?
The primer one, that's spark plug because he puts the primer in there, starts the whole thing.
Okay.
That one's Pookie, just for fun.
Which one?
DNA polymers.
Is Pookie? Because why?
Just for fun.
It builds you up.
Yeah, it builds character.
So wouldn't that be like Jet then? Because doesn't he always? Okay. Whatever helps you remember. Jet is Pookie's husband, the one who's always like, "Ah, come here, Pookie, you look beautiful."
Who the heck's what are you watching?
Lastly, helicase.
Fly.
That's fly.
Like
That's
the fly in the
yeah.
zipper.
Yeah.
It unzips it.
Some are great. Fly is funny. Spark plug's funny. Pookie, clearly there's some like you need to get on the same page with it.
Wait, what TV show though are you watching?
It's on TikTok.
Oh, we don't watch that.
It's for the bet. It'll rot your brain right out. Tell you what.
That's right.
Okay, does anyone else have any good nicknames that they want to share in our last minute of class? Helicase is the welder. Ligase is the welder. That's a great one. Yeah, does anyone else have any good ones? Okay, as you leave, if you have any, let me know. Study up for Wednesday if you want to boost your grade. Thanks, you too. You know what, Dane? I think I will make it optional. I'll make it optional. But I haven't posted any of your grades yet, so you're really risking it.